And so thank you once again for the invitation to speak. We're really thrilled to be present today and take your questions and tell you about the project, which has been running just over 1 year. And so the project's called HYDROSENSING, and what we're trying to do is to discover how plants sense water stress. So there's one missing person today, Elon Shani, who's the corresponding PI. Unfortunately, couldn't join us, and I'll explain his role within the project briefly. So the project is very much about using a synergy funding to support addressing this question. Imaging is very much at the heart of this of our approach, as is genetics and structural biology. And Christine and Thorsten are going to really elaborate more about this. In terms of our collaboration, just to give you a little bit of background, Elon and I have worked together a lot over the past years. The collaboration with Thorsten and Christine is very new. I'll explain at the end of my introduction why we felt the need to reach out to colleagues in other disciplines to address this very big question. So just to talk a little bit about water, we're all aware water is rarely an unlimited resource for plants. We see this beautiful picture on the left in the mangrove forests where water is clearly in excess. But as you can see on the right, the majority of the time water, or at least the form of water, is not ideal for plant growth or is limiting. And so plants have to adapt to periods of water stress. And if we think about water stress globally, it's actually the largest single reason for crop losses. Over the 10-year period, it generated a total loss of $280 billion from 2008 to 2018. And drought was by far the biggest reason for these losses, over a third of those losses. I read a recent report last week about Europe this spring. All of us are very aware we've had a very dry spring, and this has led to losses in the EU alone this year of close to 28 million euros, half of which is down to water stress. Water stress and climate change is obviously going to only get worse with changing climate conditions and precipitation patterns. And as you can see, removal of water from ground sources is going to be exacerbated, particularly in this central belt of countries, many of which include countries in North Africa and Europe, close to home. Within Europe itself, soil moisture levels have in recent years been at historic lows. In 2022, there was the 500 years worst drought. Many of you remember how the rivers such as the Danube and the Rhine were incredibly low. And the picture on the right shows the levels of soil moisture. And you can see how not only agricultural, but also natural ecosystems will be stressed, highly stressed under these conditions. So it leads to the question, what does actually water look like in soil? And given the audience, given you're interested in imaging, I thought I'd show this movie. This is taken using CT imaging. I'll tell you a little bit about this. This is a soil sample taken from our experimental farm at Nottingham. And you can see here the soil particles. You can see the blue, which has been added with false coloring. And then you can see the air spaces. And as you can see, the soil is incredibly complex. However, in a moment, we're going to fly through this matrix and what I call the root cam view. So imagine you have a camera at the end of your root. This is what a root sees when it's flying through the soil. So essentially it's growing through, and you can see how the water is distributed on the surface of the soil particles. There's a lot of air in there. A typical soil, when it's fully hydrated at field capacity, you can have 25% water, 25% air, and 50% solids. So this is what a root would see under normal hydrated conditions. And we did this work. Given the audience today, I just put this slide in just to illustrate a lot of our CT work is done through the Hounsfield facility. Hounsfield was the Nobel Prize winning inventor of CT. He was born locally to Nottingham, and the family very kindly agreed that we could name our facility about him. Our facility is dedicated to plant-based phenotyping, plant and soil phenotyping, and we have a number of different imaging systems that go from tiny egg cup sized imaging objects up to entire columns. And I can talk a little bit more about that if you're interested at the end. So we've done a lot of CT imaging over the years. This is one of our first papers we published over a decade ago. And this illustrates how roots can change their shape to adapt and capture water more efficiently. So as you can see here, we have on the left-hand side a root growing, and these are identical plants genetically. One has been grown within a column of moist soil. You can see it forms lateral roots all the way around the circumference, whereas one is growing down a pore, and you can see the roots sense there's only water available on one side of the root and changes its shape. So this really illustrates how plant roots really are shapeshifters, and they are able to adapt their shape to maximize resource capture. So obviously when water's limiting, we're all aware that plants will have to adapt their physiology to deal with water stress. And so this initial reduction in soil moisture is sensed in the root. Then there is a process of interorgan signaling, and this triggers a number of different effects. For example, closing of the stomata is an obvious one, which we're all aware of, to reduce moisture loss. And this is mediated through a number of signals, which have been increasingly studied in recent years. A great deal of progress has been made in terms of sulfate, for example, being an important signal, peptide signals moving from the root to the shoot. And so this is not a focus of our project itself. We're very much more interested in this very initial sensing mechanism. How do roots initially sense water stress, rather than this interorgan signaling, which generates this cascade of intertissue signaling events? So the way we actually are studying water stress is to use a process called zero branching. And this is a mechanism where if a root grows through an air gap, for example, as we've illustrated here in this CT image, you can see how the root, when it's growing in the upper soil column, it branches happily. As soon as the roots then enter this air gap, this is about a 2 centimeter air gap, you can see how branching completely stops. And then as soon as it re-enters the lower soil column, you can see how branching resumes. So we have a lovely response here that we've used to study how plants respond to local water stress. And to cut a long story short, we've studied the molecular mechanisms that underpin this. I am not going to show lots and lots of data today as an introduction. I'm going to show you some artistic representations. If you're interested in this topic, please read the paper. There are about 50 supplementary figures in this paper. Feel free to browse, but I'm just going to summarize it today. So if you imagine a root is growing through in contact with moisture, how do plants sense the moisture is available? They could measure this actually directly through sensing water, or do they do it indirectly? And what we discover in this report in this paper is they do this indirectly. They actually, what they do, the root essentially senses the co-movement of a signal, in this case auxin, the hormone auxin, with water. So they don't measure water directly, they measure it indirectly through the co-movement of the hormone signal auxin. And they do this by placing auxin in the outermost tissue, and as water is taken up by the plant and moves through the plasmodesmata radially inwards, it will co-move auxin with it. And the auxin moves to the innermost tissues, including the pericycle, which then triggers lateral root branching. And that's how plants sense whether water is available within the soil. It's very simple. It's purely based on physics and the co-movement of water with auxin. And the question is, what happens when the root then goes into an air gap where external water is no longer available? And this is a question which has intrigued us. And I'd like to illustrate how we have simplified this. We've taken it out of soil and we've grown plants within an agar system, and we get roots to grow across. And we've looked at the dynamic changes in water movement, and we've simulated this using hydraulic models developed by Valentin Couvret at UCL Leuven in Belgium. And what we discovered is that when roots lose contact with external water, they essentially rely on their internal sources of water, and the water then moves outwards from the phloem and effectively makes up for the difference of the lack of the external water source. And then this allows the plant to adapt and to continue to grow across, in this case, the air gap. Now, in addition to the phloem providing water, what we've observed using biosensors for hormones such as ABA, you can see here that as the root grows across the air gap, we actually can detect the release of ABA. This is this water stress hormone. And you can see here how ABA is increasing. This is a FRET-based sensor developed by our collaborators in Cambridge, Xander Jones's team. And you can see that as the root grows across this air gap, there's an increasing levels of ABA detected in this zone here behind the root tip. And that as soon as the root then touches the water source and returns to its normal state, you can see how the ABA levels completely change during this recovery period. So this illustrates an incredibly dynamic behavior. And so what we observe then is that in the dry zone, we see this complete change in the direction of water movement. It goes from, rather than inwards when there's an external source of water, to an outwards flow of water. And in addition to the flow of water from the phloem, you also get release of ABA. And this triggers a series of responses, adaptive responses, including closing the plasmodesmata. And this effectively shuts down auxin movement inwards and shuts down branching. So in effect, the root doesn't sense water directly. It actually senses it indirectly through the redistribution of what we call hydrosignals, auxin and ABA. Now, this model, which we describe in this paper, raises some interesting questions. Now, all of you are aware that phloem is constantly being downloaded, but ABA is only released during water stress. So there must be some form of gating of ABA release in response to water stress. And this work, I should acknowledge, a fantastic researcher, Poonam Mehra, who was the first author and co-corresponding author on this work. And she's done an absolute outstanding work, and she's continuing to characterize this process with a new ERC starter grant. She's a real rising star, someone to watch. Now, through work that Poonam and our Israeli colleagues have done, they've identified that ABA is actually released from phloem companion cells. And it appears the phloem companion cells almost act like water stress sensing tissues, and which essentially will, under the right conditions, release ABA. And the question is, how on earth is this doing it? And this is really the crux of what we want to address in the hydrosignaling project. So essentially, we've come up with a very simplistic model in which the gating of the release of ABA from the phloem is essentially controlled through changes in the volume of this or another cell type, which then triggers a signaling cascade, which then triggers the release, the activation of the ABA efflux carrier and the release of ABA and triggering the adaptive responses. Now, we hypothesize that receptor kinases are involved, and this is a key reason for Thorsten's involvement. And also, there are other steps we're likely to be involved in the ABA release and through this osmoregulation response. So, to address this question that Elon and I originally have worked on, we realized that we couldn't really address this question, this mechanism, without bringing in experts that operate at very different scales to either of our groups. So Elon is very much a geneticist and a physiologist. I'm a root phenomist, and I'm very interested in systems biology, but I don't operate at the cell wall scale, neither does Elon. And similarly, we do not work at the structural biology scale. Much of the mechanisms which are likely to emerge for water stress sensing are likely to be operating at the molecular scale through the molecular components. And so this is why we wanted to assemble the HYDROSENSING team to really cover a far greater range than was present with Elon and our teams. And so we needed to develop multi-scale synergies. And this is really where the team really coalesced to adopt a multi-scale approach which spans the nanoscale to the macro scale to really address this question. So I'm going to stop here and hand over now to Thorsten. Okay. Is that okay? And I'll hand over to Thorsten and probably take questions at the end.
Yeah, absolutely okay, Malcolm. Although you hand over to me.
Sorry, apologies.
But it's not a problem at all. Yeah, so we are quite flexible in our team. This is, I think, one of the nice things. Yeah, so hi, my name is Christine Ziegler, and I'm the structural biologist in the team. And although I come from crystallography, I have switched, as many others, to the fantastic method of cryo-EM single particle analysis. And most of you know that this technique, like now 12 years ago, have revolutionized structural biology and allows you now to get atomistic 3D structures from proteins, also in a very complex content, to very good resolutions, targets which are for a long time most likely not were able to be crystallized. So I would like to go through that really in a nutshell. It should not be the topic of my talk to talk about that, because I'm fully aware structural biology is per se not really a bioimaging method. Nevertheless, Malcolm already pointed out it is an important part also to understand what we see, for instance, in the phenotypes and in the CTs. So what you see here is a workflow which I've stolen from Alex Sobolewski's really nice structure determination of a glutamate receptor from Arabidopsis, which demonstrate quite nicely why cryo-EM single particle is so straightforward, because you need the protein. Yeah, so we work on protein basis. It has to be of high quality, but it has not to be much. Yeah, it has to be a quite high concentration, but not huge amounts like for crystallography. And then the critical step is that you do what we call a vitrification step, in which you trap the protein in the same native states it is there in the solution, and then you collect with a highly automated cryo-EM more than 10,000 of micrographs. They look very contrastless, but what you see here at these little dots are really the proteins, how they would swim around in the protein solution. And then you do just an image processing step in which you cut out out these particles. You find those which are similar. You align like 2,000, 5,000 particles, and this reduces a noise so far that you get quite a lot of nice atomistic details already in projection. And then by Fourier space reconstruction methods, you can put these different orientations back into a 3D volume, which can happen quite fast when the protein is there. With the high automated microscopes, you can have such a structure ideally like in 3-4 days. Yeah, and this is the idea. No? So this is what makes the method so attractive, and this is also the method we try to implement here in our hydrosensing consortium. We have such an automated electron microscope in Regensburg. It is something which you see here already unpacked. Normally it looks just like a cupboard, and I just mention it because in Regensburg we are one of the few groups who as a group operate such a machine. It's not a core facility. We really operate it by our group. They have a lot of fun, as you see here, and that helps us also to stick to projects which are perhaps a little bit more challenging. And what is so challenging, especially with the hydrosensing project, I will try to explain you in the next 10 minutes. So Malcom already explained quite nicely that we want to go into the heart on a molecular basis. What are in fact the initial events when the cell encounters drought stress? And there are different locations the cell can do that. And this is definitely something we will work our way through during the project. But today I want just to focus here on this interesting and very characteristic cell wall plasma membrane interface, what you see here in the cartoon. And this is kind of the same cartoon Malcom showed you, where you have like different proteins, intermembrane proteins, which are kind of in contact also with the cell wall, and the output signal is then an ABA transport. So what are we interested in, to break that down a little bit, is definitely from drought stress, you get a mechanical signal which comes from the altered water availability. And now the question is, when we want to investigate that on a structural basis, how we mimic that on a molecular level. Yeah, so on a protein level, how we can mimic such a physical complex part. And also when we talk about mechanical stress, how much is it and what kind of stress? And this is something where the consortium is so strong, and Thorsten will talk about that, I guess, a little bit, that this is an information we need to know to really come up with a realistic in vitro study on a structural basis. The next point, and this comes to the function of these proteins, is that at some point they have to sense, they have to understand this is a mechanical signal that threatens the cell, and this will not be black and white, it will be gradually. So they have kind of to perceive first the mechanical signal and then transduce it in some kind of reaction. So one of our tasks is now, by knowing the structure, knowing the function, to identify sensors. And this will not again be like black and white. There will not be one sensor. There will be most likely an array of sensors and a more integrated signal. And I come to that point a little bit later. So identification of the sensor complexes by also showing that they, during activation, undergo a conformational change. And the last point, which is also on the structural biology part, is now we have sensors and they receive a signal, they get activated, and they do this to now transduce this signal in something else the cell can understand. This can be a biochemical signal or an electrical signal, but it is intrinsic part of their function. And that is something what we try to understand by really coming up with a molecular movie of their action, stress sensing, perception and transduction. And this would mean we need to solve now structures of these proteins in a chronological order to understand what's really happening when the process gets into action. And I think there's no better way than cryo-EM to do that, single particle analysis cryo-EM. Also, we have to deal with a couple of challenges in this specific project. So first of all, stress conditions. Let's think about what kind of mechanical stress we have here. Due to the a very unique architecture of a plant cell, where the plasma membrane is fixed to the cell wall at adhesion points, every change in water availability will lead to different forces at different locations. This is an interesting part to look at, and for sure plasmolysis is an extreme example, but you can imagine that even if you have degrees of drought stress, the different sensors will sense this here the most. There's the most changes. And this is a point we have now to think about. Well, we have these tensions now due to drought stress, but how we mimic that in a structure. We have an isolated protein, how we can mimic this, yeah? And this is something where the community, not me alone, but a lot of my colleagues also think a lot for membrane proteins. Well, the best option would be to insert them in proteoliposomes. Then you could perhaps try to manipulate them. But what is used in the most cases is the next best, which are nanodisc. These are in fact little small lipid bilayers which are surrounded by a scaffold protein or a polymer. And this allow now to, by altering, for instance, lipid composition, lipid length, adding things to that, to really try to change physical properties of the lipid bilayer. And I brought you a nice example from a mechanosensitive channel, which was incorporated in a nanodisc. And here you can, by use, for instance, of cyclodextrins, where you kind of delipidate, yeah, you could create an open state. So you could activate by a signal which comes from the membrane, and you can then go from a closed to an open state and can investigate that and can link that then, for instance, to studies you do also in the proteoliposome system. You see here this channel sitting nicely in the proteoliposome. So this is something which is part of my work, to find here the best reconstitution system based on the results I get also from Thorsten's group to mimic now the mechanical stress. Now, the other point is, this was point one, we have the transduction into a chemical reaction. And here we have a little bit the problem that in plants we have a quite overlapping situation. So as you know, drought stress can also kind of induce osmotic stress, or high salinity can result in mimicking conditions. So here there will be a large part of our work to think about which are the unambiguous signatures of these different stress kinds. We are not that far already, but I want to show you how important this is for a structural biology experiment. And I will go to show you how you can use this then by using another structural biology example from my past. I'm coming from the osmotic stress field for bacteria, and I have investigated for a long time an osmoregulated beta-in transporter, which at one side is activated by internal potassium concentration. You see here the beta-in uptake rate in dependence on the potassium concentration. And you see here quite nicely that in this window between 200 and 300 millimolar of internal potassium, the transporter really goes up in activity. But it's not the only signal it needs. That brings us again to overlapping signature. It also needs a signal which comes from the membrane stress. stress, yeah, from differences in membrane, physical membrane state. So you get full activation only if you add potassium, but also here by adding tetracycline, you get here a membrane stress, and then you get full activation. And if you know this, and this is where we have to come to, you can use, in fact, now structural biology to investigate that in detail. So here you have the fully down-regulated state of this transporter from cryo-EM. You have the osmosensory unit, this is the C-terminal helix, which makes contact with an adjacent protomer. And here is the upregulated state, where this interaction is inhibited. The C-terminal domains find each other and liberate now the transporter, which kind of bend a little bit upstairs. That is kind of nice to see, but reality is completely, so reality also encompasses a lot of transition states, which tells you a lot about the mechanism. And for this BET-P, we could solve now with cryo-EM different structures in which different transition states were reached. You see here always the osmosensor, which have different conformations. And if you put that together, you can now get like a 2D map where you can see what happens if you change, for instance, potassium, when you go from down-regulated in a transient state and an up-regulated state. And this is more or less what is our plan also for the targets, the hydrosensing targets in the project, which we will, together with Aylon Shani, who can, by using really fascinating CRISPR-Cas techniques, can be identified quite precisely based on phenotyping. So now in the last part, I will just tell you what are a little bit the limits here, yeah, because this is very nice, but I told you we need it chronologically. So the question is, when happens what? And that's the way why you want to go into a molecular movie. And just to demonstrate that, I want to show you here what would happen if we just keep going in the conventional way. If I showed you that we have a protein, we have a ligand, we have conformational changes, and then we have a kind of end product. And if we freeze that together with our ligand, our substrate, we would get a distribution of different states. And you see the transient states here, yeah, would be not populated. And this would give you a completely different picture. But if you now go to the fourth dimension and do time-resolved cryo-EM by using now methods to kind of stop the process in between, you can then also investigate the transient states, which give you then the chronology and the molecular movie. So there are different ways to do that. I have this from a very nice a review from David Hasselhoff. And here is two parts which I want to focus on. You could either use the possibility to vitrify the protein, irradiate this, and then with a caged substrate, you can kind of start a reaction. This is in the microsecond possible. The other possibility is here this flash photolysis, and I would take some time to tell you how we do that in Regensburg. So we have a flash photolysis setup built up now here. And this helps now to just irradiate just before freezing to start a reaction where you can populate these intermediate states. And you can do that with a commercial vitrification robot, which you have to modify a little bit, but you can do that with high power LEDs. You see that you can just put them here in the humidity chamber, and that allows you, in fact, doing monitoring changes as fast as in the millisecond range. This is exactly the time you need when you do vitrification and plunge freeze here the sample. So this is something wait, sorry, I have to put my pointer here back. So this is what we see here in my movie. Unfortunately, I can start it here. So we see it here starting, and here we can now put in a time for irradiation before plunging. Yeah, so this is for a slower process where we are in the second range, but we can also use a method where we just then plunge now, where we irradiate while the grid is plunged down. So what do we expect here? So you see it here, yeah? So now what we want to do with that is that we have now synchronized different states, but in reality, it will not be 100%. That means you will still have a mixture. And here we want to use AI-based processing where we can separate now the different populations and automatically create now 3D volumes from that. If we would do that by hand, there are just too many possibilities, but we can use machine learning options and then go automatically to different classes and just scroll through them and decide then from the reconstitution quality about the different states. And this is something we have implemented already for proof of principle systems, and we would like, especially for the hydrogens, which can be switched by lipid interaction, and every mechanical signal is most likely triggered by lipid interactions, and bring that into place and get done the molecular movies of our different hydrogens. So with that, I would be at the end. I hope I could give you some kind of overview how that can function. And I will now give over to Thorsten to wrap the story up.
Okay, I'm not sure I can wrap up the story, but I can talk a little bit about the tools that we are developing and some imaging technology that we are using. And so first of all, I'd like to actually show the people which do the work, because I'm not spending much time in the lab. So the key person is Luis Alonso Bess here in the background, who has really done a tremendous job at establishing the technology that I'm going to talk about at NTNU. So how did this start out? Originally, my interest is in cell wall integrity maintenance. So the idea being that plants are monitoring the state of their cell walls and initiating adaptive responses if the functional integrity is impaired, which happens, for example, during exposure to biotic or abiotic stress. A couple of years ago, we published a paper which then kind of was condensed down into this model, where we are highlighting how composition and structure of cell walls and cell wall damage affect cell wall integrity, CWI. And the receptor kinase, DCS1, is essentially monitoring cell wall integrity and initiating, if necessary, adaptive responses, which involve different phytohormones like jasmonic acid or, for example, abscisic acid, which Malcom already mentioned. And we've shown that actually interactions between the cell wall and the plasma membrane are responsible for the production of abscisic acid. And abscisic acid supposedly modulates turgor pressure, which is significantly higher in the plant cells than in animal cells. And this in turn supposedly modulates, together with DCS1, the mechanical characteristics of cell walls. And so I think a couple of the key points here are that turgor pressure and cell wall mechanics are interconnected, and that cell wall integrity signaling and involving jasmonic acid and ABA signaling coordinated downstream responses. And that took us to this point where we got involved in the ERC Synergy Grant application, which was successful. And the key point or contribution of my group is the development of imaging technologies. So we are currently working on thread-based sensors to detect plasma membrane cell wall displacement. We are using molecular rotors to look at viscosity, which have been developed by Joris Sprakel's lab in Wageningen. We are trying to develop an in vivo turgor sensor, which is quite challenging, politely phrased. But most importantly, we are also developing and working with real microscopy and some microfluidics. And this is what I'd like to talk about today. So when we think about analyzing mechanical characteristics of biological materials, there have been quite a few different methodologies developed over the years. And I'm just giving you here a bit of random selection, I guess. to illustrate the variability observed. So, for example, the most common one is atomic force microscopy, which gives you a readout in the form of information about the Young's modulus. However, for example, this particular technology is mostly in vitro and only kind of useful for the surface of organisms or tissues. We have other technologies, image analysis-based technologies, elastography techniques, or body P, essentially a film-based methodology which allows you to investigate the viscosity levels in the tissue. The technology which has caught our eyes, which we think is quite exciting, is Prion microscopy. And the idea here is that laser light is interacting with the sound waves in the material or in the sample, like a cell, simplified here. And this interaction leads to a scattering. And this scattering is the Brion effect. And what has been observed or described is that the shifting of the Brion peak, shown here as the small peaks, can be correlated between higher stiffness levels and more shifting of the peak. Interestingly, second readout of this measurement is the width of the peak, which allows you to draw conclusions about the viscosity in the sample that you are analyzing. Now, the interesting thing, or there are several arguments or reasons why I find Brion microscopy is so interesting, and these are kind of listed here. So you can actually access deep tissue layers below the surface. It's non-contact, non-invasive, it's label-free. You have a subcellular resolution, and the nice thing is it's actually compatible with other imaging technologies. So, for example, you can combine the Brion measurements with fluorescent detect proteins in the same tissue. And what is Brion microscopy actually measuring? And this is summarized here. So the frequency shift is related to the elastic inelastic light scattering. And the readout is the longitudinal modulus. And the interesting thing is for us that the frequency shift, which is fairly straightforward to detect, is proportional to the longitudinal modulus. And I think it's important to stress at this point that the longitudinal modulus is a different readout than what you get with the atomic force microscope. So with the atomic force microscope, you are measuring the Young's modulus, shown here in orange, whereas the longitudinal modulus is actually also a mechanical characteristic or property, but it's a different readout. So most of the time, if you see some change with the Young's modulus, you also see a change with the longitudinal modulus, but you don't have to always see the same differences. When we now look at the technical setup, so normally the idea of the Brion microscope is that you have the Brion module essentially involving a Viper spectrometer, which is connected to a bulk standard confocal microscope. In our case, we've created a custom built Viper or Brion module and hooked it up to a Leica SP8. And the nice thing is that you then get as readouts or outputs wide field images, you get stiffness, and you get fluorescence. Now, when we look at the resolution we are looking at, so here you see on the y-axis the spatial resolution in micrometers, and on the x-axis the acquisition time in seconds. And the different dots, or different colored dots, are essentially highlighting slightly different technical setups for the Brion microscope. But I think one of the key messages is, for example, that in our setup, the lower resolution limit is approximately 1 micrometer. And if we measure 1,000 pixels or images size of 1,000 pixels, we are measuring approximately 10 to 12 minutes. So what do we actually get when we start measuring? Our model system are rhodopsis seedling roots. And what I'm showing you here are some results which Louis has produced, that we are trying to understand how actually the stiffness of the rhodopsis seedling root looks like in 2 dimensions. So we use seedling roots which are expressing a plasma membrane marker, and these are essentially different optical sections along the root in different kind of depths. top, closer to the middle, further down. Now you can do the same images with beyond microscopy. And there, essentially, the heat map gives you an idea where the stiffer areas are in red and the less stiff areas are in blue. Like, for example, these outside areas here are the watery solution. What you now can do is you can actually measure along the axis here, highlighted in orange, and trace or track the individual cell layers. This is why we use our ceiling roots, because they are highly organized and exhibiting highly reproducible patterns. And so what you can see here is on the y-axis, the frequency shift, and on the x-axis, the actual kind of distance along the route. And the interesting thing here is that you can see that the developmental age is changing, and we can track this for the individual cell layers. So the stele is the central cell layer, and then you have the endodermis, cortex, and epidermis is the most outward layer. And interestingly, I think the two key messages, or three key messages, which can be taken away here is that the stiffness levels seem to change in the individual tissue layers while they are kind of developing and differentiating. That apparently the stele has the highest level of stiffness, whereas the epidermis seems to have in this area the lowest stiffness level, with cortex and endodermis being intermediate. But when we approach the terminally differentiation state, they seem to be very similar levels, not much difference between cortex, endodermis, and epidermis, whereas the stele is still distinct. And in a way that is kind of expected, because the stele has reinforced cell walls to support water transport. We can do the same exercise looking at the viscosity. And there again, the stele is quite distinctly different the whole time. It does change to some extent, whereas the other cell layers are similar but not identical, with the endodermis exhibiting elevated viscosity levels. Now, this is a very, I would say, rough resolution. You can't really tell much about individual cells. So what we did do in the next step was to actually look at an individual cell layer, the cortex cell layer, and track the changes in cell shape for the cortex cells. And I'm highlighting here the three areas which we looked at, like very early, later in development or differentiation, and then very late. And what you can very nicely see here in the meristematic region, there the cell shapes are quite distinct. Then in transition zone, the cells get larger, they're still square. But in the elongation zone, they have elongated particularly in one direction. Now, what you can do is you can, for individual measurements, actually compare the stiffness for the longitudinal cell wall versus the radial cell wall. And what you see is that actually the stiffness of these cell walls is changing. So initially, there's no significant difference in the meristematic region. Then in the transition zone, you can see that the stiffness of the radial cell wall is going up and seems to be further or more pronounced in the elongation zone. This is kind of nicely fitting with the elongation, or with the elongation, expansion of the cells along the root growth direction. You can also look at the viscosity, and there the images already give you the indication that we have different effects. So in the meristematic region, there's actually a difference in the viscosity between the different cell walls, which is still there in the transition zone, but in the elongation zone, this seems to be gone. And I think it's also important to stress here, we have a resolution of 1 micrometer, which allows us to actually look at individual cell walls and see if there are particular areas which are, for example, have a particular level of viscosity, but also changes in the stiffness, or particular areas which are particularly stiff. Okay, so this is the developmental perspective. Now what we can also do is we can actually look across the different cell layers. And I'm showing you here in the images from where we use biomicroscopy to look at the across the different cell layers, highlighted here by this orange rectangle. And this is the data for the stiffness, and this is for the viscosity. And we have quantified this and done this multiple times. And so you see here for the cell walls, the stiffness in blue and the viscosity in red. You can see that, again, the outer epidermal cell wall seems to be the softest or less stiff, and the stiffness levels go up, with the steel cell walls, or the pericycle steel cell wall, exhibiting the highest level of stiffness. Viscosity behaves similar, but not identical. When we now look at the cytoplasm, there's a more gradual change. So you have epidermal cell layer, cortex, endodermis, and steel. You can very nicely differentiate how, for example, cortex and endodermis have very similar viscosity levels, which are distinctly different compared to epidermis. Stiffness to steel. And viscosity here is less pronounced, only the steel is showing distinct differences. Now, what we also wanted to understand is how actually manipulating cell wall metabolism may affect stiffness of cell walls and viscosity. And we've used different mutants, which give resistance to a particular drug that we are frequently using, which modify the structure of pectin, particular compound in primary cell wall signaling, and also ABA production. And what we actually find when we look at the individual kind of cell walls, that, for example, the XXT1/2 double mutants do exhibit profound significant differences in cell wall stiffness. And similarly, you have effects with the TCOS1 kinase. When we look at the viscosity, there we actually don't really find profound differences. And I think this just shows us that particular cell wall components, which are required, for example, for pectin formation or xylosyl production, do affect the stiffness, whereas others don't really have profound effects. Now, to summarize, I hope at least I have shown you some interesting results which highlight how we can use biomicroscopy to investigate both stiffness changes and viscosity changes in living tissues, which is, I think, a big step forward compared to previous methods, which were either very indirect or in vitro. And we hope that by kind of being able to investigate stiffness and viscosity, we can contribute to the success of the ERC project by actually understanding what happens on the cellular level when plants experience drought stress and try to adapt to a changing environment. Thanks for your attention.